Hello everyone. Well, up until this point, we've been taking a look at Mendelian genetics and the crosses of Gregor Mendel, taking a look at all of the different crosses that he did, looking at the various different traits in pea plants. And what we have seen is that every single one of those traits that Gregor Mendel did take a look at are referred to as a dominant recessive relationship. So for each one of those genes, whether it is for the seed shape or the pod color or the height of the stem, there was one single gene for each one of those different traits. And for each gene, there were two different possible alleles, and one of the two alleles was always dominant over the other one. That's one possibility, but it's not the only possibility. So in this lesson, we're going to talk about, well, a couple of other possibilities: incomplete dominance, codominance, and what are referred to as multiple alleles. So this first one here, incomplete dominance. Incomplete meaning that we don't have one allele which is dominant over another allele. I did mention that prior to the time of Gregor Mendel, what most people did believe in was blending inheritance. So when we think of blending inheritance, that is really what incomplete dominance is. So with the blending, what we see is that we have the two homozygous forms that have a distinct phenotype. But when we do have the heterozygote form, we have a completely different phenotype, a phenotype that is an intermediate between the two different homozygous forms and is clearly distinguishable from either one of those homozygous forms. So if we take a look at the picture that we see here, three different flower colors, and the three different colors that we see are the red color, the white color, and the pink color. So hopefully it's fairly obvious that we do have an intermediate form here. And quite often, if you do think of incomplete dominance, it's very similar to what you would see in art, where you're mixing colors together. So if we do take red and mix it together with white, what we get is the intermediate form that is going to be the pink. So think about this whenever you come across information about a cross. Ask yourself the question, do we have a third different phenotype that not only is different, but it's kind of a blending and quite often related to colors? So think about, well, this one here, we have red and white, we mix them together, the intermediate form is pink. If you take blue and yellow and mix them together, you end up with green. If you take red and blue and mix them together, you get purple. If you take black and white and mix them together, you get gray. And I better stop there because that's about the extent of my knowledge as far as the visual arts. But yeah, think about sort of the blending as this intermediate form, the mixing together, and quite often kind of the mixing together of different colors. So this example here that we're taking a look at with the cross in snapdragons, the flower snapdragons, I'm showing you here the first row, the parent phenotypes. So we have a red flowered snapdragon, which is crossed with a white flowered snapdragon. I give you the F1 generation, but unlike what we saw with any of Gregor Mendel's garden peas, we don't see one of the phenotypes in the F1 generation that we saw as a parent phenotype. We don't see the red, we don't see the white, but in fact we do see, again, that intermediate, the pink color in the F1 generation. So clearly this is not what we call a dominant recessive relationship. That was the case with Gregor Mendel's garden peas. That was not the case here. We also said a second indication that one trait is dominant over the other is that we're using capital and lowercase letters, because a capital letter does indicate that that trait is going to be dominant. Notice that I'm not using capital and lowercase letters here. I'm using, well, all capital R's. I'm just distinguishing between the two different alleles by using either a 1 or a 2. So in this case, the R1 would be the allele for red. The R2 would be the allele for white. The gene that we're talking about here would be the flower color gene. So for this flower color gene, we have two different alleles, the R1 allele, the R2 allele. One is not dominant over the other one, so I am not, and I cannot, in fact, use capital and lowercase letters. So as soon as you determine that it is not a dominant and recessive relationship, you should not be using capital and lowercase letters. You need to choose some other way to indicate the difference between the two different or more than two different alleles that you might be talking about. But from that point, once we do come up with the symbols that we're going to be using for the different alleles, it's very similar to what we have already seen with our crosses that do show a dominant recessive relationship. So from parent number 1, which is the red color, they have, of course, because they are diploid, they have two copies of this gene, as all diploid cells do have to have. So two copies, one on each of the chromosomes. So when the gametes are formed from the red flowered snapdragon, those gametes will only have one chromosome, so they will only have one of these R1s. So from the red parent, that one will be contributed. That's the only thing that can be contributed from the red flowered parent because that is the only version of the gene or allele that they have. From parent number 2, all they have are the R2 alleles, so that's all they can contribute. So what we would then see is 100% of the offspring in the F1 generation, all of them are going to be pink. So if we are writing the phenotypic and the genotypic ratio, it is the same in this case. So we could simply say 100% are going to be pink, and they are going to be R1, R2 for their genotype. So now if we take those offspring from the F1 generation and we cross them, so now what I have here, this is going to be the parents. They were the offspring in the F1. Now they're the parents, so I'm just going to call them the P2, the parents in the next generation. So we have our one parent that's going to be heterozygous, and the other one, of course, is going to be exactly the same, will also be heterozygous. So when we set up our Punnett square across the top, we're going to have the gametes from our one parent, R1 and R2, and along the side from the other parent also R1, R2. And if we go ahead and fill in our Punnett square, R1, 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 R2, and I'm just going to continue to write the R1s first here to be consistent, and then finally the R2, R2 that we do have in our last square. So what this does say is based on probability, out of the 4 squares that we have, one of them will be homozygous for the R1 allele, 2 of them will be heterozygous, and the last one that we have here is going to be homozygous for the R2 allele. So once again, the genotypic and the phenotypic ratios in the case of incomplete dominance, they will be the same. So this is our ratio that we have here. I'll put a P for the phenotypic ratio or the phenotypes, and I'll put a G for the genotypic ratio or the genotypes. So what we have is a 1 to 2 to 1, both phenotypic and genotypic ratio in the F2 generation. So that is incomplete dominance, and most of the time this is a typical example that you would see is taking a look at flower color. We can also have incomplete dominance in humans, and one nice example of this is with hair. So whether hair is curly, wavy, or straight hair, that is determined by one single gene, of which there are 2 different versions. Again, one is not dominant over the other, so we can't use a capital and a lowercase letter. We are talking about hair, so why don't I just use an H? And why don't I say that I'm going to use just a capital H on its own to represent curly, and I'm going to use, again, a capital H with a little prime to represent the straight. So the intermediate form in this case is then going to be the wavy. So at the left-hand side, the picture on the left-hand side, this is going to be curly. So those individuals will have two copies of the capital H. On the right-hand side, the straight hair will be two copies of the H prime, and the intermediate form, it's going to be H prime, generating the wavy phenotype. Now, of course, we have many, many more different hair colors than simply black, brown, red, and blonde, all kinds of intermediates in between, and that can be for several different reasons. There may, in fact, be more than one gene that is responsible for hair color, and there can also be more than two alleles or even three alleles for a given gene. So when we are talking about multiple alleles, it is for a single gene, but there could be more than two possible versions. So in the case of Gregor Mendel's garden peas, it was always just two different possible alleles. So if you think about the flower color, it was either the purple or the white color. If we think about the seed shape, it was either the round or the wrinkled, and those were because of two and only two different alleles. But in the case of hair color, there are many, many different alleles, and depending upon the combination that an individual gets of those two alleles, that will determine the hair color that they do have. So kind of keep in mind as well, even though there may be 10 different alleles for a single gene, each individual is diploid and will only have two copies of the alleles for that gene. So a nice example of multiple alleles that it does have in your textbook as well is taking a look at the coat color in rabbits, and this one is referred to as the C series, as you can see here, because they are using the letter C to represent the different alleles that we have. You might notice here that there are capital C's and there's also a lowercase c. So this one does in fact show a dominant recessive relationship, but on top of the dominant recessive relationship, what we also have are more than two alleles, so we do have multiple alleles. So the different coat colors that are represented here, this is what is referred to as the agouti coat color, this is the Himalayan, this is the chinchilla, and this one here is the albino. So the four different possible alleles are the capital C with a superscripted H, a capital C with a CH, and a lowercase c. So I have written them in this order for a reason. The one at the far right-hand side is the one that is the most recessive. The one at the far left-hand side is the one that is the most dominant. And the other two fit somewhere in between, and in fact they do fit in this order. So how do we know that the lowercase c is the most recessive and that is going to produce the albino? Well, the only way that you can get the albino color if there are two lowercase c's. That is the only possibility. What about for the capital C's? Well, it doesn't matter what you put the capital C with. If you put it with a capital C superscripted H or the superscripted CH, it still generates the agouti coloration. So that means that the capital C is going to be dominant over the capital C with either the superscripted H or the CH. If we take a look at the Himalayan, if we don't have the capital C on its own, but we have the other combinations, it doesn't matter what we put that C superscripted H with, we will always generate the Himalayan. So Himalayan then is going to be dominant over the two remaining ones. And what fits in between the Himalayan and the albino is going to be the chinchilla. So this one here is what is referred to as a dominance hierarchy. So we have one single gene that is for the coat color in the rabbits. But we have various different possibilities because each parent will only get two copies of these alleles. From this dominance hierarchy, we can then generate the four different phenotypes of agouti, Himalayan, chinchilla, and the albino. So once again, that is referred to as a dominance hierarchy. And you can only have that if you do have multiple alleles. The last dominance pattern that we'll take a look at here is called codominance. This is a little bit different than incomplete dominance. Once again, it is not a dominant recessive relationship. One allele is not dominant over the other one, but it's also not a blending. So if you think about on a team, if you have co-captains, you don't have one captain that's overshadowing the other one. That would be a dominant recessive relationship. You don't have their individual traits that are kind of blended together. That would be incomplete dominance. Instead, you have each one individually contributing their own part. So that's kind of what we see with codominance in terms of genes. So both alleles of a gene, both of them are expressed as an observable phenotype. So let's take a look at a few examples here. So in cattle, we have a couple of colors that I show at the top here. The one at the left, this one is in fact white. The one at the right is referred to as red. So if I write this down here, this is the one here that is white. This is the one here that is red. So this is not incomplete dominance. If it were incomplete dominance, then we would get a pink cow, but we don't have the blending together. Instead, what we see is this cow down below. This cow down below is referred to as roan. So this one here, no, it's not pink, but what we do see is there is some white hair and there is some red hair up around the head and the neck area. So we see both of the phenotypes that were present in the homozygous forms, the white and the red, that are both visible in the heterozygous form. So the roan in this case is going to be the heterozygote. If we take a look at the flower on the right-hand side, this is completely different than the snapdragon that we took a look at. We don't see pink. Instead, what we see is a flower that has both red and white. If this were incomplete dominance, if we took a red flower and a white flower, it would end up being pink. But it's not. This is codominance. So if you take a red and a white flower, what you get is an F1 generation flower where you see both of those colors that were present in the homozygous form. We see on the flower a distinct red color, and we also see the distinct white color. And we see a similar pattern with this last example that I give you here with the fish, where we have the two different homozygous forms, the red and the blue, and the F1 generation. The heterozygous form is not purple, but it is blue and red. Both of those colors are visible in the heterozygous form. That is codominance. Really nice example to talk about here with human blood typing. So this one is kind of nice because with human blood typing, we do have several different things going on. We do have a dominant recessive relationship. We do have multiple alleles, and we also have codominance. Not incomplete dominance, but codominance. So in this case here, you're probably aware from Biology 20, or maybe even before that, that there are four different basic blood types. They are blood type A, blood type B, blood type AB, and the last one, the most common one, is blood type O. The different alleles, there are four different blood types, but there are three different alleles. And this one here, where these symbols come from, it is a capital I that is used, well, a capital I and a lowercase I. And what this does stand for is for isoagglutinogen. And the three different ones that we have are the capital I with the A, the capital I with the B, and a lowercase I. So it should be pretty clear that I'm using capital and lowercase letters, and because capital does imply dominance, lowercase does imply recessive, that means that this one here, the lowercase I, is going to be recessive to both of the capital I's. The other two, these ones here, are the ones that show codominance. In terms of the phenotypes, there are a number of different possibilities. I'll write all of these down. So the first possibility is we can have homozygous for the IA. we can have the IA with the lowercase I. For these two here, they both have the capital I, and because the capital I is dominant over the lowercase I, that means that both of these genotypes will generate the same phenotype, and that is going to be blood type A. The next couple, we can have the IB with the IB. We can have the IB with the lowercase I, and very similar to what we saw with the blood type A, the capital I is going to be dominant over the lowercase I, so both of these genotypes will give the same phenotype, which is the blood type B. The next one, if we take the IA and the IB, this is the one that's going to show the codominance. So this is not a blending, it's not a dominant recessive relationship, this is codominance. So both the information for the IA is going to be expressed on the surface of red blood cells, and for the IB expressed on the surface of red blood cells, and these are the individuals that have blood type AB. So they do not have a mixture of blood type A, some of the red blood cells being blood type A, and some of the red blood cells being blood type B. That is not the case. Every single one of their red blood cells is going to be similar because it is going to have the characteristics resulting from both the IA and the IB allele. The last one that we have here is if we have two lowercase i's. In this case, there is no information for the IA or the IB, and that is the one that results in blood type O. So in terms of the phenotype, what is the actual phenotype if someone does have blood type A, blood type B, blood type AB, or blood type O? Well, what we do talk about is what is on the surface of the red blood cells, and what is on the surface sticking out of the red blood cells. It's actually a glycoprotein, a protein associated with a sugar, and there is a different glycoprotein generated from the IA information from that allele compared to the IB information. There is no glycoprotein generated from the lowercase I. So if we do have someone that is blood type A, what that means is on the surface of their red blood cells, they have something that is very distinct, a glycoprotein, and the instructions for making that glycoprotein come if someone does have this allele here. So whether they have one copy of it, whether they have two copies of it, it doesn't matter. If they do have the IA on every single one of their red blood cells, they will have this antigen or this glycoprotein. Someone that is blood type B, they have something different sticking out of their red blood cells, a different glycoprotein that looks a little bit different. So this results in a different phenotype. The phenotype is the blood type, resulting from the different glycoprotein or the different antigen, and anyone that has the information, the allele for blood type, the IA allele, or sorry, the IB allele is going to produce this B glycoprotein or the B antigen. So if someone is blood type AB, again, they don't have a mixture of these two. That is not the case. What they do have on every single one of their red blood cells is they do have this one here. So that was the A glycoprotein. And they also have these ones here. And those are the B glycoprotein. So how were they able to make both the A and the B glycoprotein? Well, they had to have had that allele, that information to make that glycoprotein, so they have to have the IA allele, and they also have to have the information to make the B glycoprotein or antigen, so they need to have the IA allele as well. Someone that is blood type O, well, they don't have either the IA or the IB, so they don't make either of those glycoproteins. So on the surface, they don't have either of these glycoproteins that are sticking out.